So just to recap on what we were discussing in the previous class. So the body has the ability to defend itself in two ways. So it has something called your general defense system, and these include the non-specific methods used to prevent the entrance of pathogens into the body and the killing of those that do. Okay, so it's split up into two different kind of levels. So you have your primary level defense, and that includes things like the following that are listed here. So we have the skin is obviously a major barrier. It's the biggest barrier that exists in our bodies to prevent pathogens from entering your skin or to your body. If your skin cracks, then that can be a method through which pathogens actually enter into the body. Mucus in the nose and cilia in the nose as well, they help to trap specific pathogens and emit them from the body as well. Your second level of defense then, it consists of things like your phagocytic white blood cells. Bigger versions of these white blood cells are known as macrophages. Complement proteins as well, which work in tandem with the phagocytic white blood cells, and an inflammatory reaction, which is something similar to your fever that will be experienced. So this diagram here that I have circled is a pretty good representation of how the phagocytes actually work. So as you can see here, they're pseudopodia, so they're quite similar to your amoeba. So your pseudopodia here, they actually form around a bacterial cell. So you can see the bacteria or whatever pathogen it is, they're located there. And then finally, that actually closes up, and you can see there the bacteria are engulfed and they're ingested or they are digested. And ultimately, that is how they are removed from the body. If you come back here then to your defense proteins, they help to destroy viruses and other pathogens. Okay, so that is listed just here. Those are examples of complement proteins. And then interferons, they're another set of defense proteins, and they prevent viral multiplication, and they help to limit the spread of virus infections such as colds and influenza. Your inflammation then, that includes things like your fever. Okay, so fever, chills, they are all examples of inflammation. And generally speaking, a higher temperature causes bacteria and viruses to slow down their reproduction. Okay, so that is how a fever works to our advantage. So the next thing we're going to look at now is the specific defense system. And that defense system, it attacks specific pathogens. It works by the production of antibodies, and white blood cells are also used that destroy body cells that have been infected with a particular pathogen. Okay, so white blood cells destroy body cells that have been infected with a particular pathogen. Okay, so we have two types of white blood cell that mainly carry out this function. So they are known as monocytes and lymphocytes. Okay, so we've already come across a type of white blood cell in the form of the phagocytes. Okay, and now this is two more types of white blood cell that are called monocytes and lymphocytes, but they are only associated with this specific defense system. So monocytes and lymphocytes are two types of white blood cells, or leukocytes, and both monocytes and lymphocytes are formed in bone marrow. Okay, so I have that highlighted on your slides. I'll revert back to them in a few minutes. So once they are produced, these cells move away from the bone marrow and into blood vessels and into other parts of the lymphatic system, such as lymph vessels, lymph nodes, and the spleen, and also the thymus gland in the chest. So once they leave the bone marrow, they move to these 4 different locations. So monocytes and lymphocytes react to pathogens in different ways. Okay, so they both have their own specific type of function depending on what the body actually requires. So monocytes develop into white blood cells called macrophages, and macrophages recognize foreign molecules that are present in the surface of pathogens. So when a macrophage digests a pathogen, antigens from the pathogen are normally displayed on the surface of the macrophage. And these antigens then stimulate the production of antibodies. Okay, so let's go through that again using this diagram here. So we have our macrophage, okay, and what's actually happening here is that the pathogen has these little antigens which are on the surface of it. And when the macrophage engulfs it or surrounds it, the little antigens that were on the surface, so you can see them here, they're little spikes. Okay, you can see the little spikes here. Those actually relocate onto outside of the macrophage that has ingested them or digested them. And as a result of that, these antigens then that are present on the surface of the macrophage then stimulate the production of antibodies. Okay, so lymphocytes, this is kind of where our antibodies come into play a small bit. So lymphocytes, they fight infection in two different ways. So some attack body cells that display antigens in their surface, and these cells may be infected with a pathogen, or they may be cancer cells. And cancer cells produce abnormal molecules that are identified as foreign by lymphocytes. Then other lymphocytes actually produce antibodies. Okay, so that's the difference between those two. Some actually attack cells that display antigens on their surface and try and get rid of them, and then other lymphocytes help to produce antibodies. Okay, so if we just come back here to our slides for a second. Okay, so our antigen there, as you can see the definition for it, if you didn't know what it was. So it is a substance that causes lymphocytes to produce antibodies. They are produced in the red bone marrow, and they're found in the spleen, in the thymus, in the lymph system, and also in the blood. I'm actually going to highlight those 4 areas as well because those are quite important. I highlighted those in the book. And antibodies then are proteins, so that's the type of biomolecule. If a question comes up asking you what type of biomolecule are antibodies, they are proteins, and they are produced by a lymphocyte in response to an antigen. Okay. So back here now again to our book. So antigens and antibodies then. So the word antigen is based on the word antibody generating. So that's actually where it comes from. It's just a mixture of the 2 words. So that might be a handy way for you guys to remember it. And antigens include molecules from the coats of viruses and the cell walls of bacteria, fungi, and other microorganisms. So they're actually coming from those specific molecules themselves. Antigens are also found on foreign cells such as pollen grains, incompatible blood transfusions, transplanted tissues or organ cells, and cancer cells either. So the antigen-antibody reaction is a highly specific reaction. So it's very much like the enzyme-substrate complex. You know how only a specific type of enzyme will work with a specific type of substrate? It is very much like that. Okay, so this means that there is a precise fit between the antigen and the antibody. Again, similar to our enzyme substrate complex. So just like enzymes and substrates, each antibody is complementary to a single antigen. So each antibody is complementary to a single antigen. So they are highly, highly specific. And each antigen stimulates the production of only one specific antibody. Okay, so now before we go any further, just let's have a look at the definitions for antigen or antibody again. So the antigen is the foreign molecule, and that stimulates production of the antibodies. And an antibody is a protein produced by white blood cells in response to a specific antigen. So our antigen, those are the bad guys, those are the foreign molecules, the pathogens that enter the body. And antibodies are what are produced in response to those in order to try and fight them off. So antibodies bind to antigens and they allow the pathogens to be destroyed by lymphocytes. So your antibodies here, if we just have a look at this diagram here. Okay, so your antibody is the Y-shaped object here, and they help to bind to the specific proteins which are located on the coat of the virus or in the, let's say, cell wall of bacteria. So they bind to those specific antigens that are located on the surface. They all have different types. For example, this one here is circular, this one here is triangular, this one here is a square shape, and you can see that the place in which it fits into is actually specific to the antibody itself and the antigen itself. So antibodies bind to antigens and they allow the pathogens to be destroyed by lymphocytes. So antibodies bind to antigens and they allow the pathogens to be destroyed by your lymphocytes. And antibodies can also trigger the complement system, which results in the pathogenic cells being burst. Okay, so antibodies trigger the complement system, which results in the pathogenic cells ultimately being burst. And when they are burst, they can no longer have an impact on the body. They cannot infect the body cells, or they cannot use the body as a host in order to replicate their DNA or RNA. So this diagram here is pretty good in terms of the relationship that exists between the antigen, which is the foreign particle, the antibody, which is produced by our body in response to the antigen, and then the lymphocytes that ultimately will digest the structure that is created by the forming of the antibody and the antigen together. Now I don't have a markup on this, so I can't draw on these slides, but in the top left hand corner up here, if you can see, there is an incorrect antigen receptor. So your antigen here in this case is the little bits that are sticking out of the green object, and your antibodies are the little blue Y-shaped structures located on the outside of the white blood cell. If there is a correct antigen receptor, you can see there how they will bind to the invader particle, and ultimately it will create this reaction which occurs. Similarly, if there is an incorrect antigen receptor, there will be no reaction. So it is very, very similar to this whole enzyme substrate complex. When they bind together, it's like a lock and key mechanism. It will work. But if the incorrect antigen or the incorrect antibody is there, then they will not be produced. Okay. So when this happens, when the antibody actually fits the antigen, it will bind with the antigen, and you can see here kind of over on the right hand side, the antibodies are actually bound to the foreign pathogen or the foreign antigen, the green object here. And once that happens, phagocytes can ultimately engulf the invaders marked with the antibodies, because they know that the body's specific defense system has marked these as they have to be getting rid of imminently, quickly, before any more of them take over any more body cells. So your phagocytes or your macrophages, whichever the case may be, they actually engulf them, but only the ones that are marked with the antibodies. Okay, so that is how antibodies work. Okay. Back here then, so we have the duration of immunity. Okay, so just another little bit to go. So your duration of immunity is all about the time after which you have been infected with a particular virus or a particular bacteria, how long afterwards you can actually be immune to that. So after an infection is fully overcome, some of the antibody producing lymphocytes remain in the body for a long time. Okay, so your antibodies really here, they are really important in your body because they ultimately offer you protection from various types of diseases or infections that you may have experienced before. Generally speaking, you will not experience them again, very generally speaking. So these long-lived cells are called memory cells. So very appropriately named, these memory cells, they last in the body. They will recognize any sort of antigens that enter the body, and there will be an automatic antibody response to them. So if a second similar antigen enters the body, these memory lymphocytes can rapidly produce large amounts of that specific antibody much more quickly. Okay, so although you may be reinfected with the same type of virus or bacteria, your body recognizes it and it's able to produce antibodies at a very, very quick rate, an exponentially quick rate, as opposed to if it was your first time experiencing the particular virus or bacterial infection or disease. So this is why we usually do not suffer from the same infection for a second time. We can suffer from colds or influenza more than once, and this is because there are many different forms of cold and flu viruses. So different forms of the same thing, what do we call them? We call them mutations, okay? So new cold and flu viruses develop by mutations, okay? So back to your cell division chapter, we talked about mutations, about how they're different forms essentially of the same cell or whatever the case may be. So the thing about it is that viruses constantly and bacteria constantly change so that our body does not recognize them, and therefore there are easier targets to infect us with, essentially, because we do not recognize the foreign invader, which means that we can spread it more easily, and ultimately the virus gets to reproduce, or the bacteria gets to reproduce a lot more quickly. Whereas if it did not mutate and stayed the same, then all types of viruses and diseases will probably be eradicated very, very quickly. But because they are living organisms, they do have the ability to mutate, just like humans also have the ability within their cells to mutate too. So on first contact with an antigen or a pathogen, the body takes about 14 days to produce the maximum number of antibodies. Okay, so what's that reminding you of now? It should be reminding you of all the advice going around about coronavirus or COVID-19. Oftentimes it can take up to 2 weeks for the first symptoms to actually display themselves. So this is kind of being aligned here with your own body's natural inflammatory response to produce antibodies. So on subsequent contact with the same antigen, antibodies are produced in large numbers in about 5 days. Okay, so about a third of the time. And this prevents the reinfection from producing symptoms. So although you may have been reinfected with the same bacteria or the same virus, you probably will not display any symptoms because your body has been really, really, really quick in order to catch it before it multiplies. Okay. So this table here sums it up pretty well. So you have your first line of defense. I'll just change the highlighter color here. So you have your first line of defense, which is considered to be like the skin, your mucous membranes, secretions from the skin, and the mucous membranes. Okay, so those are all things that we came across in our previous lesson. Your second line of defense includes phagocytes, defense proteins, and inflammation. Okay, so those are all part of the general defense system. And then your specific immune system is your third line of defense. Your monocytes, they destroy antigens, and also your antibodies as well. And these work in tandem with each other. So your antibodies usually attach to your antigens, and then they are digested then by things like your monocytes and lymphocytes. Okay. So the next thing we are going to be looking at is induced immunity. Now, before we move on to that, just to revise on what we have gone through again today. So we started off here on this slide. So you have about immunity, what an antigen is, your lymphocytes, where they're located, what are antibodies, how antibodies and antigens, how they actually work together, what induced immunity is. So induced immunity is when the body contains specific antibodies against particular antigens. So those are like your memory cells, okay? And then you have this diagram here finally, which sums up the entire thing, okay? So I'm going to leave it at that for this class, and then your next class then is all going to be about induced immunity and also vaccination and immunization, and then we'll see how we're getting on from there. Okay, again, any questions, please drop them into the Google Doc, or anything that I've left unclear, okay?